this video, we will talk about what are the direct methods for gene transfer when we talk about transgenic technology in plants. Okay, so now we will begin. First, to begin with, we will talk about what is transgenic technology. So, transgenic technology is a method when we're going to transfer genetic material from one organism to another organism, and the organism that we're going to create will be called as genetically modified organism. The gene that or the segment of the DNA that is introduced into the genome will be called as a transgene, and the branch of biology that deals with these transgenic organisms are called as transgenics. Now, here, what we are going to create are plants that have desirable characteristics that are taken from different sources. So, this process we call it as transformation, and the manipulation of genes in case of in organisms is called as genetic engineering. Now, talking about the overview of the production of transgenic plants, we have gene identification and isolation. So first we have to select the gene of interest from our source, and then we have to isolate it. So isolation can be done by using molecular biology techniques such as PCR. We can do gene cloning. So in gene cloning, we're going to amplify the gene of interest so that we have enough amount of it to introduce into the host. So in gene cloning, we have methods such as PCR, where we're going to create multiple copies of the target gene. Next, we have gene insertion. So gene insertion can be done by multiple methods. One is gene gun, microinjection, electroporation. We also have agrobacterium mediated transformation. So in today's video, we will be discussing in detail about the direct methods of gene insertion into plant cells. Now, after we have inserted the gene into the plant cells, they will be selected based on the uptake of the genetic material, and they will be regenerated. Whatever plants are created will be screened and verified, and they can be commercialized or field tested in order to produce the particular plant with a desirable trait in amplified amounts. So we will start talking about the methods of gene transfer, specifically the direct methods of gene transfer in plants. So here we are going to take the foreign DNA and directly introduce it into the plant cells. When we use another vector, when we use another intermediate molecule, we will call it as indirect method. But in the direct method, we'll take the piece of DNA that we want to transform, and then we will give it directly into the plant cell. So this relies on the physical and chemical means. We are going to make the cell wall permeable to these pieces of DNA so that they can be taken up by the cells and they can get the required characteristics. So we're going to talk about four methods in this video. First one is electroporation, particle bombardment, microinjection, and polyethylene glycol method. So we will start with electroporation. In electroporation, short electrical pulses are created, are subjected to the cell that creates temporary pores on the plasma membrane or the protoplast. So we will take our plant cell and then we will subject it to short electrical pulses. The short electrical pulses can be given directly to the plant cell, or mostly we will be giving it to protoplast. So what are protoplast? Protoplast are naked cells that have lost their cell wall. We will supply these electrical currents, which is then going to create pores that can take up my foreign DNA. So we will prepare the protoplast. We can remove the plant cells by using enzymes such as cellulase or pectinase. We can mix the DNA along with the cells. We will subject the cell to electrical pulses. So here they have mentioned the conditions: 250 to 3000 volt per centimeter, and temporarily is going to create disruptions in the membrane. The DNA which is now present in the surrounding or the extracellular region can now enter into the cell through these pores. After the DNA has entered, we will do the membrane resealing. After the pulse, the membrane will reseal because of the fluidity of the membrane, and then the protoplasts are regenerated by plant tissue culture to create new cells. So the applications of electroporation, we can use it for plants like rice, maize, tobacco, wheat, and soya bean. It is mostly used for monocots. The advantages of this method is that it is simple and efficient. You don't need any vector or a bacteria, and wide range of plant species can be used. But the limitation is what we are going to use is a protoplast culture. The protoplast culture is difficult in some species because we cannot remove the cell wall for all of the species of plant cells. And we have cell viability that can be reduced because we are subjecting the cells to shock. So these are the limitations. Now moving to the next method, we have particle bombardment, gene gun or ballistic method. So what we're going to do in this method is we're going to take our gene of interest and then we're going to coat it on metal molecules. These metals are then shot into the cell which pass through the cell wall and enter into the nucleus. So we're going to shoot the DNA coated microparticles. The coated microparticles are usually gold or tungsten. So we take gold or tungsten and coat it with our gene of interest, and then we are going to shoot it into the cell. So first we're going to prepare the microprojectiles. Gold or tungsten is then taken. We will coat it with the DNA that we want to introduce, and then the coating is done using calcium chloride and spermidine, which can help the DNA adhere to these microprojectiles. We will load the gun and then we will accelerate it. We are going to create the pressure using helium gas, and which is going to forcefully pressurize these ballistic molecules into the cell. Next is bombardment. We fire it into the plant tissue and then the DNA delivery happens. The DNA is now present inside the cell close to the nucleus, or sometimes we can also aim into the nucleus where they can directly be present next to the DNA of the host cell. Now, once it is released, it is integrated into the plant genome. It can be via non-homologous recombination. We will select the plants that are showing the desired traits and then we will regenerate the successfully transformed plant cells. The applications of this method, it is used for both monocots and dicots. It is suitable for also chloroplast transformation. So we also have DNA in the chloroplast. The chloroplast DNA can be transformed using ballistic methods. This is the method that is used in golden rice and BT crop. The advantages of this method is that any plant tissue can be transformed by using ballistics. It can be used to transform cells, embryo, as well as intact tissues. It allows the transformation of organelles. As we've studied, chloroplasts can be transformed by this method, and transformation efficiency is more. So more number of cells turn up getting up the piece of DNA that we want to introduce into their genome. The limitation is that ballistic method is an expensive method. It can cause tissue damage because we're shooting random particles into it and they will insert multiple times in the genome. Now moving to the next method, which is microinjection. We have the direct injection of the DNA into the nucleus or the cytoplasm using a fine glass micropipette. So we will take our cell of interest and we will introduce the gene of interest directly into the cytoplasm or the nucleus using a very small needle or a micropipette. So first one, we are going to prepare the protoplast. We will isolate the protoplast using enzymes such as cellulase or pectinase, and we will keep them on immobilized surfaces such as agar or a slide. Then we are going to start the injection. We can use a micro manipulator or a micro capillary needle, which is going to, the DNA solution is going to be present inside this needle. Next, we're going to start the injection. In the injection process, we're going to take the ultra thin micropipette needle into the cell through the cell wall. Here we have removed the cell wall. So we're going to move it through the plasma membrane and go into the nucleus or the cytoplasm and release release the DNA. The DNA that has been released will now be integrated into the host DNA. By the process of recombination, we can have the recovery and regeneration of the selected hybrid cells. So whatever cells have been taken up the gene of interest will be called as transform cells. We will be selecting the transform cells and then regenerating them. The applications, we can use this method for introducing DNA as well as RNA or even proteins into the cells. We can study gene expression as well as subcellular localization using this method. It can be used for somatic cell genetics as well as cytoplasmic manipulation. Now, what are the advantages? The method is very, very controlled. So the delivery is directly into the nucleus. It can deliver multiple molecules It can also deliver organelles, and we do not need any vector. The limitations are the technicality is very difficult. It takes a lot of skill to develop the ability to hold a single cell and inject the DNA into the nucleus. The outcome is a very low throughput, which means we cannot do a lot of samples through this method. Only one cell at a time. It is costly, and then the protoplasts that we are isolating or the large cells are often limited. We cannot isolate protoplasts for all the species of plant cells. Now, the last method that we will be studying is polyethyl glycol mediated method. So polyethylene glycol is a chemical fusogen, which means it is going to enable the DNA to it's going to enable the cells to take up the DNA by making the plasma membrane temporarily permeable. So it's going to convert the plasma membrane into a permeable structure so that they can take up the DNA that we're providing them. So first we're going to isolate the protoplast, and then we're going to mix the DNA that we want to insert into the with the protoplast and in the presence of calcium ions. So the calcium ions have the ability to go and bind to the negatively charged membrane and then neutralize it. So once the cell membrane has been neutralized, we're going to apply PEG to it. So it acts as a fusogen, and it is going to cause the aggregation of protoplast and fusion of DNA to the membranes. So once the DNA fuses to the membranes, the DNA is taken up by the membrane and entering into the cytoplasm. So in the last step, we're going to remove the PEG, is washed off so that PEG is not remaining in the cells, because PEG can cause toxicity, and the protoplast are then recovered to regenerate into transformed plants. So what we have here, we have the plant cells which were taken off the cell wall to create protoplasts. The protoplasts are then mixed along with the gene of interest and PEG in the presence of calcium ions, which are going to neutralize the cell membranes, and the PEG is going to allow the fusing of the DNA to the plasma membrane, and which is going to further result in uptake of the DNA, which can be integrated into the host gene. This method is used for tobacco, rice, wheat, maize, potato, and Arabidopsis. So this is a method for stable transformation. The advantages, it is very simple, it is inexpensive. We can use it for a wide range of species, and no specialized equipment such as the electroporator or the micro injector. All these specialized instruments that we studied before are not used here. The limitation is it only works on protoplast. Protoplast can be very fragile, and transformation efficiency is very, very low. Sometimes the PEG that we are adding can be remaining in the cell media itself, which is going to create toxicity when it is present in high concentration. And the DNA integration is very, very random. It is not going to happen in the location that we want it. So with this, we complete the methods. I have also given a comparative table for the four direct methods of gene transfer in creation of transgenic plants under the genetic engineering. So now we have transgenic technology as a method for creating plants with desirable traits. For transferring the gene of interest to the plant, we have four direct methods, which we have learned in this video, which is electroporation, particle bombardment, microinjection, and PEG mediated transfer. So thank you so much for listening.